We all went through school. We all heard about the rods and the cones. That's how we see things. The rods and cones take that information, and then the occipital cortex turns it into a visual image. But about 10 years ago, it was discovered that there's this third type of receptor, and these receptors project to other parts of the brain.
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 175th episode of this podcast dedicated to shining light on the optimal workings of your own brain by any and all means at your disposal, including, it turns out, light. In this episode, we're going to be looking at sleep cycles, circadian rhythms, mood, sleep deprivation, and even the fact that your skull can operate a little bit like a pair of sunglasses, which is a really weird and probably very little known fact that I had no idea of until I did this interview. So a lot of different topics covered in this one. It's a bit of a potpourri, but a really interesting one with two experts, a research scientist by the name of Dr. Scott Killgore and a Finnish venture capitalist by the name of Timo Ahopelto, who is involved with a product called the Human Charger, which actually applies light directly to the brain via the ears, which, counterintuitive though that may be, has actually been shown to have scientific efficacy. If you hang around until the very end of the episode, I'm going to tell you about a recent study that very much calls into question the just say no doctrine of Nancy Reagan. Yes, it's true. That's a 30-year-old doctrine, but it still has a lot of echoes in our current culture. Hang around for a partial refutation in the ruthless listener retention gimmick. But as usual, let's kick things off first with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So there's two types of people in the world. There's the type who don't know what the amygdala is, and there's the type that think that the amygdala is the part of the brain related to fear responses and fight or flight, generally dealing with bad situations. And according to a new study at MIT, just published in the last issue of the journal Neuron, it may be that neither of these groups actually knows much about the amygdala. So there was a study last year conducted in the lab of Dr. Susumu Tonegawa, the director of the Riken MIT Center for Neural Circuit Genetics, and that study was looking at two distinct populations of neurons in a part of the brain known as the basolateral amygdala. These two populations are genetically programmed to encode either fearful or happy memories. And in a new study, they were seeking to further clarify the connections within these two neuron populations and to determine the functions of central amygdala cells that receive information from the basolateral amygdala. The researchers wound up dividing the neurons into 7 groups based on where they were physically located and also the genes that they were expressing. Then using optogenetics, which is a technique that we've talked about a lot, where brain cells are directly triggered using light, they triggered each of these neuron populations to see what they would do. And in 5 of the 7 cases, the mice in the studies, this was a mouse study, when they were triggered with light, they wound up exhibiting behaviors that would get them more light. In other words, the scientists were seeing a reward-seeking behavior. They were finding reward circuits in the amygdala, not what they expected, says Tonegawa. Classically, people have generalized the central amygdala as a fear-related structure. They think it's involved in anxiety and fear-related responses. However, it looks like the structure as a whole seems mainly to participate in appetitive behaviors. The cells that we have identified so far represent more than 90% of the central amygdala. If there are some other cells for negative behavior, it's a small fraction. In another surprising finding, the researchers discovered that the fear-linked neurons that they did identify in the central amygdala do not send messages directly to the part of the brain that they believed received fear-related inputs. That part of the brain, called the periaqueductal gray, is located in the brainstem and is involved in dealing with pain, stress, and external threats. It might be that the negative cells identified in the central amygdala get to the periaqueductal gray eventually following a circuitous route, but as of yet they don't know, and Tonegawa's lab is still trying to trace these circuits to find out where signals from the negative cells eventually lead. Continuing studies that Tonegawa's lab is doing into the basolateral amygdala are looking at its role in fear extinction, which is the process of rewriting fearful memories so they no longer have as much emotional impact. This, not surprisingly, could be used in therapies for post-traumatic stress disorder.
Smart Drug Smarts, the podcast so smart, we have smart in our title, twice.
Picked up a couple of 5-star reviews on iTunes. Listener Sid Chopra from Australia says, very informative. Your objectivity in determining the risks and benefits of supplements and methodologies is refreshing. And listener Kickstand Chris from the USA says, This is the absolute best podcast out there for nootropics and cognitive optimization. I am a registered nurse, former Air Force medic and EMT, and active in CrossFit and mixed martial arts. And what you've done is given me everything that I've wanted to research on a silver platter. Well, thank you so very much for that. Chris also goes on to say the editing and pace of the podcast is spot on, and I'm buying my first bottle of Nexus today. I mentioned both of those for a couple of reasons. First of which is that I definitely am aware that I am a fast talker and an aggressive editor when it comes to audio, cleaning up pauses, things like that. I don't like that stuff. Occasionally, somebody will write in and say, hey, Jesse, can't you guys just play a full uncut interview, press record, press stop when you're done, give us everything in between? And the truth is, I actually encourage my editor to not do that, to go exactly the opposite direction. If I let things drag with 5 minutes of an episode of ums and ahs and pauses and fillers, and over the next couple of weeks, tens of thousands of people listen to the episode, then I've wasted, at very least conservatively, thousands of hours of other people's time, which I don't feel good about. So the pacing is a deliberate choice. Hopefully that's cause for celebration among most of you out there. Definitely a case where you can't please all the people all the time, but I gotta go with my gut on that one. And as for the second comment by Kickstand Chris, he made reference to the nootropic stack Nexus, so I will too. Nexus, as you probably know, is one of the two stacks that we've got over at axonlabs.io. Axon Labs is the retail end of things over here on Smart Drug Smarts. I got a phone call from my dad yesterday saying, hey, Jesse, you should be aware the company Taser, which makes tasers that cops tase people with, they just changed their name to Axon. Axon is how they're listed on the New York Stock Exchange now. I don't know if this is good or bad, but it's probably something you should know about. But yeah, I guess I should take this opportunity to formally acknowledge that there is no relationship, implied or otherwise, between Axon Labs and our nootropic stacks and the taser weapons that are used to subdue people who need subduing. Both us and the company formerly known as Taser got their inspiration from the axon, which is a part of a neuron, the long spindly bit where the magic happens, the message actually gets transmitted. If you imagine a neuron as a potato gun, it's the long shaft that the potato shoots down. But back to the point, both Nexus and Mitogen, its sibling stack, are available over at axonlabs.io. And yeah, thank you very much to both Kickstand Chris and Sid Chopra for taking the time to write those iTunes reviews. Very, very much appreciated. As are those of you who have subscribed to the Brain Breakfast newsletter, which is also something you can pass around to other friends, loved ones, literate folks, a sort of a written companion to Smart Drug Smarts, and also a little mini guide to the stuff that we're researching as we come up with episode topics. You can sign yourself up for the Brain Breakfast at smartdrugsmarts.com/newsletter. Last but not least, following the double interview that you're about to hear, I'm going to tell you about a product giveaway that we're doing. We actually have two human chargers. That is the light-emitting brain gadget that we're going to be talking about in part of the coming interview. And we're going to be giving those away to a couple of lucky listeners, one of whom could be you. I'll have all the details on that contest, how it works, what you need to do following the main interview. A little background on the human charger, what this thing is. I've had one of these myself for several months now. It's really tiny. It's almost like having a couple of sticks of gum in your pocket, but it's got a little single-button stick that's attached to a cord which has two LED earbuds going into your ears. You press a button, the light goes on both so you can see it in your hand and also the light goes on within your ears. There's a little silent countdown for 12 minutes. You feel a faint heat within your ears and 12 minutes later you're done and you put it away until tomorrow. So a really simple little gizmo, and you'll hear more about it in the coming interview, why it does what it does. But I wanted to give you a mental image in your head so you could picture this thing as we're talking about it later.
Smart Drug Smarts.
Okay, so as mentioned, this is a double interviewee episode. I'm getting really fond of this format. It allows us to juxtapose things in what I feel like is a pretty interesting way. Not that 2 is the magic number. Sometimes we might have 3 or more people. But in this case, we had a couple of interviews that I didn't want to delay unnecessarily based on the fact that we publish weekly. And I felt like they had enough overlapping or at least tangentially related topics that we could probably weave something together here. Another thing that this allowed me to do is one of the 2 interviewees, Timo Ahopelto, is a venture capitalist behind the company that makes the human charger product. We're mostly talking science in the interview, but of course he does have a financial stake in the product that we're talking about, and I never want the episode to have the feeling that it's a long-form commercial or anything like that. But sometimes people that are involved in these brain products from the business end of things still have really interesting things to say and are worthy interviews. And I feel like the multi-interviewee format of having maybe somebody from the science side and somebody from the business side, intercut those together as appropriate, is kind of a nice way of learning about the science and also seeing what's out there in the marketplace as far as ways to apply this. In any event, Dr. Scott Killgore is part of the Division of Sleep Medicine at Harvard Medical School, where he is an associate professor of psychology, as well as director of SCAN, that's the Social, Cognitive, and Affective Neuroscience Lab. He's been involved in a huge number of sleep-related studies, and we'll be talking about a lot of those, but particularly focusing on his work on the interactions between sleep and light. As we all know, a flashlight in the eyes is not a great way to fall asleep, but it turns out that science has gotten very, very detailed, especially in recent years, looking at specific means of how light can be used to optimize our sleep and wake sleep-wake cycles. And Timo Ahopelto from the company Valky is based out of Finland, a country in a far northern latitude, where the amount of sunlight on offer can be very, very short, especially during winter months. And he'll be talking about the development of the human charger, which uses the application of light to deal with a lot of the issues that Dr. Scott Killgore talks about. One last thing before we get into this interview. At one point, Dr. Scott Killgore is talking about emotional decision-making under the effects of sleep deprivation, and he explains the trolley problem, that classic moral decision-making, what do you do kind of bad scenario that I won't even go into here because you probably already know what it is and have heard about it a million times, including on past episodes of this show, and because he explains it fully in the coming interview. But this was kind of one of those judgment calls where, is there a way of truncating this so people who already know what it is don't get bored hearing it explained for the umpteenth time? I wound up leaving the whole thing in there, but I'm going to have a little insertion. Basically, I'm going to come in and say, hey, if you want to fast forward a minute here and not have this thing re-explained that you already know what it is, now is the time to forward a minute. So be ready for that. Dr. Scott Killgore is the one with the conspicuously American accent if you're trying to identify who's who. So with all that as preamble, let's jump in with Dr. Scott Killgore and Timo Ahopelto.
So I joined the Army, and the first assignment that I got was in a sleep lab. And I really didn't know anything about sleep, but they had me working in a lab where they were doing experiments of total sleep deprivation. They were keeping people up, sometimes up to 88 hours with no sleep at all.
What is the methodology for that, by the way? I imagine medieval torture devices, but it's probably something else.
It's actually fairly simple. We just had a room with research assistants that would stay there on shift. And so they go for about 8-hour shifts, and they just keep watching the participant as they're in the room, and they just never let them sleep. And if they start getting sleepy, they have to stand up, walk around the room, keep watching some TV, play some games, get something to eat, but they're not allowed to leave the room. They're not allowed to exercise. They just basically stay there and stay awake for 2 to 3 days at a time. Basically, they were doing studies looking at simple reaction time and what they call psychomotor vigilance, how well you can stay focused on a very boring spot on a screen and respond every time that point changes in some way. And it really didn't go beyond that for a lot of the previous studies. What I was most interested in was emotional processing and executive functions and these higher level cognitive abilities that people have. And they had really not been explored at that time to see how sleep deprivation might affect those. So we wanted to start taking a look at what happens to the brain and cognition when a person is sleep deprived, and how does that affect your higher level judgments, your decisions, your willingness to engage in risky behaviors and moral decision-making processes.
88 hours or up to 88 hours, that's a heck of a long time. Was the degradation somewhat linear, or what sort of variations were there?
Well, what we know in sleep deprivation is that your sleep pressure, or the biological drive for sleep, is overlaid on top of what's called your circadian rhythm of alertness. So when you combine those together, you get sort of this downward rhythmic pattern that gets worse and worse in a linear way, but it increases and decreases throughout the day. So at times when your circadian rhythm is actually in the upswing, you may be very close to your normal level of performance, but overnight, when your circadian rhythm starts to drop again, you see this dramatic drop in your overall cognitive abilities. But what we know about sleep deprivation is that your sense of sleepiness essentially plateaus at a certain point. So you don't feel any more sleepy after about one night of sleep deprivation.
That's interesting.
Yeah, you continue to get worse, which is what's the scary part about it, is your performance continues to degrade, but you don't feel any sleepier. And so your judgment changes. You don't know that you're in such an impaired situation, and you go for 2 days without sleep. You're a lot worse than you were at 1 day without sleep, but you feel about the same. And so you don't know that you're getting worse and that you're at a higher risk of having a car accident or making some kind of mistake.
Originally, this was an invention by 2 Finnish scientists. The other one was more an engineer, and the other one was more on physiology. And on an animal side, studying mice and pigeons and birds and even fish, it's a well-known fact that you actually don't need to have light into your eyes to have the typical refreshing effect that light has. It's enough to get it somewhere in your body or to the area of your head. And this was a kind of a pondering question in a way that, could this work in humans? People have been altering the circadian rhythm of birds by projecting light into their head instead of projecting light into their eyes only. And that's a very well-known phenomenon in animal physiology. Nobody has taken it to humans. And then there was one Nokia engineer in Oulu, which is very north in Finland. The sun never gets up there during the winter time, so it's very dark. And then he was totally depressed during the winter time. And he contacted the other guy and said that, hey, now this this is enough, so I'm going to illuminate my whole house with bright lights, and it's going to be very expensive, and it's going to be a big project. And the other guy said that, hey, don't do it. Let's try to illuminate your head and then see what happens. And then the Nokia engineer thought that, hey, people are so used to wearing all sorts of earplugs, listening to music and having podcasts and everything, that maybe what if we project light via ear canal inside of the head? And the physiology guy said that, hey, that's a great news, because basically your ear canal is like a direct hole inside of your head or into your head. And then they started testing, and then they realized being refreshed themselves. They did a couple of more devices, and this was 2007, 2008. They gave them to their friends, and the friends more energized during this northern, very dark winter. And then they started to do more like a control tests and clinical trials, and figured out that there must be something in this. And then, of course, packaging it into iPod-like device. I mean, iPod was very big at that time when they put together the first device.
Yeah.
It kind of seemed pretty obvious in a way. And it's very counterintuitive, like you should be listening to light via ear canals. So that was the starting point. And then it was an immediate aha moment kind of a thing in a way, that this is so counterintuitive, and at the same time scientifically, and I'm thinking about what has been learned about the animal physiology, it's so natural that there needs to be something. And that was the starting point.
In looking at the effects of sleep deprivation on cognition, what did you find about how that hurts us? I imagine that there's lots of detrimental effects, but it might be different maybe in some types of brain activities than others.
The ones we were most interested in had to do more with the emotional side of things. And what we found was that when people are deprived of sleep, which is no big surprise in a sense, but people's ability to use their emotions to make good decisions actually starts to become impaired. So they show a decrease on standard measures of emotional intelligence. So we actually had an IQ test that our subjects had to take before they were sleep deprived. And then when they were sleep deprived, we found that there was a significant decline in their overall ability to use their emotions effectively, and a decline in their overall empathy for other people. We actually gave them a very interesting task that isn't used much anymore, but it was like a cartoon task where we had them look at cartoons that had empty bubbles above the heads, and they had to fill in what they thought the person in that situation would say. And it was a frustrating situation, like the person lost the car keys and the wife is standing there and she's got a bubble above her head says, "It's a fine time to have lost the car keys." And then you have an empty bubble for the husband and you have to write in what his response would be. And basically what we found was that in that kind of a situation, people had much lower frustration tolerance and they tended to blame the other person for the situations much more. They didn't do well emotionally in those kinds of situations. They really took the blame off of themselves, and it was always the other person's fault.
Were there any aspects of cognition that weren't that adversely affected by sleep deprivation that did surprisingly well?
Well, what's interesting is the area that we were most interested in is higher order cognition, like executive functions, the ability to plan and sequence and think several steps into the future. And sometimes we thought for sure that those would show severe deficits. And amazingly, oftentimes they were quite resistant to sleep loss. So you'd go for a couple of days with no sleep, and we thought for sure people would have a hard time planning out several steps into the future or making good decisions in some ways. And in fact, did just fine on those kinds of executive function tasks, which was not what we expected, but people could do it. And some of the theories behind that is that these kinds of tasks are very interesting. It's fun to solve a problem. So if you've been sitting in a sleep lab for the last 2 days being very bored, it's actually kind of exciting to sit down and take a Wisconsin card sorting test or something else that stimulates you a little bit. And that stimulation itself may actually be enough to help you to perform well or do better than you would have otherwise. But it's short-lived. It's only for just 5 minutes or so that you can focus and do okay on those kinds of tests. But when it's a boring task, like a psychomotor vigilance task, which lasts for 10 minutes, those things are very boring and it's really hard to stay focused on those.
I saw that some of your studies dealt with introverts and extroverts, and that sleep deprivation actually affects those demographics differently. That's sort of counterintuitive. I was wondering, A, can you tell us about that? And B, what led you to even think in that direction?
What we're talking about really is individual differences in the ability to resist sleep deprivation. Interestingly, about 12, 13 years ago, there were a number of studies that came out starting to show that people differ in their ability to stay up all night. Some of us just don't need much sleep and we can function fine even with a day or two without sleep, and we don't show a lot of deficits. Others are very impaired and they show very severe deficits when they stay up all night. And what's interesting is it's a very consistent trait-like phenomenon. So within the same individual, individual, each time that you sleep deprive that person, you get a very similar response. Even if I were to sleep deprive you in February and then bring you back in March and sleep deprive you again, if you were resistant, you would be resistant both times. So it's a trait-like phenomenon. So we got to thinking maybe there are some other stable trait-like things that might predict that. And so a lot of investigators have tried to identify biomarkers. Are there things in your blood? Are there genetic components? Are there differences in brain shape or brain size or brain function? And an obvious one was to look at personality differences. Maybe some people differ in their personality traits that help them to stay awake more. The basic theory that we had was based on Hans Eysenk's theory of cortical arousal. This is a theory that extends way back many decades in personality. And his basic theory was that introverts tend to have a higher level of basal arousal to begin with. That's why they're introverts. That's why they like to go to the library. They prefer to be alone because their brain is already jacked up to begin with. They don't need more stimulation. Whereas the extrovert is actually low in their basal arousal. So they seek out external stimulation.
Right.
They need to be with other people. They need to get around other stimulation to make them feel at their optimal level of functioning. So it seemed reasonable to think that if you were an extrovert, your basal level of arousal is low to begin with. Maybe you might actually be more vulnerable to the effect of sleep loss because it will take less to drop you down to that level of falling asleep. Whereas the introvert is actually highly aroused. It's going to take further for them to move down the graph and actually fall into that state of sleep. So we tested that out. And in fact, that hypothesis was supported. What we found was that introverts were better able at sustaining performance during the first night of sleep deprivation relative to extroverts. It sort of falls apart after the second and third nights without sleep. But the first night, it seemed to hold up fairly strongly. And then we also followed it up with a second study where we're interested in whether introverts and extroverts would differ based on social enrichment or not. In other words, we gave them a party in the lab. So we brought in these introverts and these extroverts. So we screened them very thoroughly to make sure they fell into one of the two camps. And then half of the subjects got essentially what we called a party. They had the research assistants engage in very social ways with them. They played lots of games. It was very festive, very active kind of an atmosphere. Or we did the same experiment where every subject went into their own isolated room and they never had contact outside of an intercom with the research assistants. So no social interaction. They did all the same tasks. They played all the same games online, but there was no human interaction involved in the situations. And then we controlled for their overall level of activity with a wrist-worn activity monitor to make sure that it wasn't just one group being more active than the other.
Right.
And what we found was that it was actually the extroverts that were most affected by the party in a negative way. What we found is that the extroverts, it wore them out faster to be in that social situation when they were sleep deprived, that when they stayed up the next night, they dropped off much faster in their ability to sustain performance, whereas the introverts were able to sustain throughout the night, even though they'd been exposed to the party. And that was opposite what I was initially expecting to have happen, but it seemed to be a fairly robust phenomenon. Our theory about it is that actually the extroverts were more engaged and were using their prefrontal cortex in those situations a lot more. And there's a whole theory in sleep deprivation that it's called kind of a use-dependent decline, in a sense, where the more you use the area of the brain, just like a muscle, the more tired it becomes, and it starts to fall asleep easier as you go for a longer period without sleep.
If you look at the brain atlases which are out there, nobody had been studying the brain tissue and whether or not the brains are sensitive to light. It just hasn't been in the research focus. And what these guys did, they first started with the imaging study. So they put people into the dark room, eyes totally covered so that they couldn't see anything. And then they put these type of light guides into their ears. And then without the control person knowing, they switched the lights on and off. And then they did brain imaging at the same time. And they were able to show that the certain brain areas actually get activated because of the light projected onto them after like 3 to 6 minutes of exposure. And that was a big revelation in a way that, hey, the light actually penetrates the brain and it actually has effect in the human brain. So something's happening. And of course, the fact that something is happening doesn't mean that something is happening therapeutically, that it has an effect, right? If you scratch your skin, I mean, something happens, but it doesn't make you feel any better, right? So So that was the starting point. And then they started to figure out that, why is this happening? So what is there in a brain that is sensitive to light? And they actually got a very rare samples of brain tissue from cadavers. It's very rare to get that amount, like roughly like a 10 total brains that you can study. And then they did the brain staining and then found that actually the human brain is full of proteins called opsins, especially OPN3 and OPN4, which are fully sensitive to light. So they belong to the same family of proteins that you have in your eyes, in your retina, to detect light. And that was the kind of idea that, hey, if these brain areas are actually covered by these light-sensitive protein matter, there needs to be a function with them. If there weren't, in evolution, those proteins would have stopped to exist in a brain. But digging deeper into the literature, you can actually find some early studies where some medical technology companies and scientist groups have been studying that how does the light penetrate into the brain. And they're concluding that the skull is actually letting a lot of light inside of your head during the sunny day. And so it's actually a surprising amount of light is penetrating through the skull.
Wow, that's surprising.
Yeah, that's right. And in living here in the northern latitudes, the next step was then to start figuring out that does this really make people feel better during wintertime? And there's a term for this. It's called seasonal affective disorder or winter blues in the milder form. In the very northern latitudes, like 1 or 2 out of 10 people suffer from winter blues. And even in New York latitudes, it's like 1 out of 10. People just don't realize that they have it. It's like a downward mood during the winter months. And these studies were conducted with really depressed people to see if there's efficacy. And it was surprising to see that in the first studies, there were, of course, open label trials, like you typically start the development of these type of things. And between 80 to 90% of people who participated basically got rid of all the winter blues symptoms during a period of 4 to 8 weeks of treatment. And the treatment is daily 12 minutes of light. So you push one button and it gives you a 12-minute light dose.
How did you zero in on the 12-minute figure? And is that equivalent to 12 minutes of bright sunlight, or is this such concentrated light that it's giving you more than 1 minute per minute versus what you'd get from the sun?
It's a really interesting question because if you would be developing drugs, you have this dose escalation trials where you're trying to figure out that what is the right amount. If you have 10 milligrams of ibuprofen, it doesn't take you a headache. But if you have 1,000 milligrams, it's probably too much. 300 to 400 is probably enough. So you have kind of a titrated the right dose. And then here, what these scientists in the university were able to see is that the brain networks actually get activated after 3 to 6 minutes of light exposure. And then in some of these like early tests that were done in the university, it seemed in a way that the dosing amount between 6 to 12 minutes is the right amount. And that's why it's always a combination of art and science. What is the right dosing when you go into these sort of scientific experiments? And that's why the group selected 12 minutes. And that's where all the studies have been done. But if you look at the ear canal and you would be putting fingers into your ears, you can realize that there's actually not too much brain between where your ear canal stops on the either side. If you then look at what are the areas that are actually located in that area, there's, for example, a 3-part brain area called raphe nuclei, which is responsible partially for serotonin production. There's the substantia nigra, which is known to be for dopamine production. There's obviously the hypothalamus, which is hormonal control functions and circadian rhythms. There's all the melatonin production functions are actually almost directly where your ear canal stops. Those are the oldest brain areas, if you believe that the brain has developed evolutionary in a way that you have, depending on who you listen to, but the 2 or 3 layers of the brain. And obviously the cognitive functions are on the top and on your forehead. But real basic functions and the hormonal control, it's in the central to the old parts of the brain. And they are within few centimeters away from where the ear canal stops. So the ear canal is a really effective way to put light into those areas.
With the caveat that obviously you're the most biased person in the world to answer this question, but it seems like there's really almost no downsides to this technology. You can't overdose on sunlight per se, other than getting a sunburn. If you sprayed it out your ears for 24 minutes rather than 12 minutes, there's really no harm to be done, right?
Yeah, I know that some people are using it for more than 12 minutes. We cannot say that because we are operating under the medical device certificates. So it means that we need to stick to what we have studied. And we have studied in jet lag, we have like a 3 times 12 minutes a day. And in seasonal affective and Wiener Blutzankh, this energy increase, we have a 1 times 12 minutes a day. Some people who are like really sensitive to light, they have actually got back to us in a way that they can almost like the time by hour when they wake up compared to when they use the device. So they basically say that, hey, if I use the device like 8 p.m. in the evening, I will wake up at 6 a.m. If I use it like 9 p.m., I will wake up at 7 a.m. and so forth. It needs to affect the circadian rhythms and the cycles, how they go. So some people are that light sensitive and timed into light. So in that sense, you could also like mess up your circadian rhythm probably easily if you would take the doses here and there.
In one of these studies, we gave them a measure called the Personality Assessment Inventory, or the PAI. And essentially, it's a broad spectrum measure that looks at all different aspects of psychopathology and measures them on scales. And then we were able to measure them again when they were sleep deprived. And we found that there was an increase in levels of depression, levels of anxiety, and somatic symptoms, that these healthy, normal individuals showed this dramatic increase in those levels of psychopathology. But what's fascinating about it is that if you're actually a depressed person, if you're suffering from clinical depression, we see the opposite pattern. People who are depressed actually get feeling less depressed and may even get giddy at times when they're sleep deprived. And there's a lot of debate in the literature about that, of why is it that depression seems to have this improvement in their mood when they're sleep deprived, whereas healthy normals actually become more depressed when they're sleep deprived. And nobody's really come up with a perfect theory on that yet. We're playing with some ideas on how the prefrontal cortex may be involved in that, but it's not ready for prime time quite yet.
There is that one thing that I think probably a lot of people will be familiar with, where when you stay up way too late, you get kind of like a weird sense of humor. It only seems to be there in that one particular state. Me and my friends have called it punch drunk, but I'm sure that's not the clinical term. Do you have any insight on that?
I think it's related to the same kind of a thing. There were some studies that Matt Walker's group out in California has done, where they've shown that when people are sleep deprived, it alters the connectivity between the prefrontal cortex, which is the regulating area of the brain that regulates your emotions and evaluates emotional stimuli, and the more primitive areas like the amygdala that are involved in salience detection and emotional responses. So you get sort of a dysregulation of the emotional systems in the brain and your ability to control and regulate those. And what their studies seem to show is that it affects both positive and negative emotion. And it may come to do with whatever the stimuli are that you're being confronted with. So if you're dealing with sad pictures or scary pictures, those are going to affect you more and you're going to have some negative emotions. But if it's actually funny and you're with your friends and you're seeing something enjoyable, it may actually make you feel punch drunk and you're kind of giddy and having a good time with it. The fact is you're not able to regulate your emotions the way you are well rested.
We've talked about sleep hygiene in past episodes of ways to optimize a person's sleep. Do you feel like there's particular approaches that the public is not clued in on enough? Things that science knows that the public might not, about how to get sleep as optimized as possible?
I think the big one that we've been focusing in on is those circadian rhythms I was talking about before, and aligning that with your light exposure. A lot of times people don't think about light and how light may actually be affecting their sleep. And as I said before, your homeostatic drive for sleep, the longer you stay awake, the stronger that drive is that's pushing you to fall asleep. Just like when you haven't had water or food, you have this drive to go eat or consume water. The same thing happens with sleep, but it's overlaid on that circadian rhythm. And the big thing that affects your circadian rhythm is light. There are other factors that play into it too, but light is really probably the most powerful regulator of that. And I'm always surprised that people have not heard that much about it, but that getting light in the evening is not usually a good thing, especially blue wavelengths of light, like what you get off of your computer or your television screen or your iPad. All of those things are emitting a blue wavelength of light. And what we know about light is that light suppresses the chemical called melatonin, which is really what tells your brain that it's time to go to sleep. So if in the evening time you're sitting here looking at the computer screen for several hours, you're suppressing that chemical that normally is preparing the brain for a good night's sleep. So it's going to shift you and tell your brain that instead of being 10 o'clock at night, it's only 7 o'clock at night. So your brain's not ready to go to sleep yet. And that can really impair your sleep. So if there's one big advice I would give is to kind of turn off the smartphones and the computers earlier in the evening so that you're not being affected by that. And then on the flip side, get more exposure to blue light in the morning hours. For me, what I find is I live in Arizona now, and we get this beautiful sky in the morning. So the first thing I do every morning is I get up and I go out and I have my coffee in the morning, and I sit out for about half an hour, and I just get that big blue sky out there. And that radiant blue sky suppresses melatonin and sort of resets my circadian clock each day. And since I've been doing that, I've noticed that I sleep so much better at night when I get that early morning burst of blue light early on, 6 to 7 in the morning, and I'm more alert during the day and sleep better at night.
Now, for those of us that don't necessarily have bright, cloudless Arizona skies outside when we wake up in the morning, is blue light something that makes it through cloud cover, or is it really one of these things that if you live in a more of a Seattle-like environment, you've got to take technology to the rescue?
No, it's still enough that if you get outside, that the amount of light that is coming through the clouds is white light. So the amount of light that's coming through still has that blue wavelength embedded within it. That white light has the entire spectrum. So you're still getting the blue wavelength to suppress that melatonin. So that's still a powerful effect. May not be quite as much as a big bright blue sky, but it's still a very powerful effect if you're out in it. If you're sitting inside your living room and you don't have very big windows, that dull gray is probably not enough to really be suppressing your melatonin at the level that you'd like. But I would try to get outside if you can. Even the morning drive is for a lot of people enough to start resetting that.
How long does it take to shift your body into either melatonin production or melatonin suppression? For example, let's say you're doing everything wrong and you're in a brightly lit environment until 9 o'clock at night, but then 9 o'clock at night you go into your bedroom, it's dark, there's no light leakage. How long will it be until your body completed that switch over into melatonin production?
It takes a little while. When they do studies to look at melatonin, they usually have you sit in a room for several hours with a very dim amount of light. So they call it a dim light melatonin onset. And usually it's within a matter of hours, but it's following your circadian rhythm. So you have a rhythm of melatonin that will come on naturally if you're not in bright light. And so over the next hour or two, your melatonin will start to emerge if you're in a darkened situation. Just exposing yourself to some light can actually suppress it and shift it for up to an hour or so sometimes, just by being exposed to even a brief pulse of light sometimes.
So your team sent me a human charger, which I've been using for a couple of months now. Thank you very much. And along with it, a lot of the marketing materials deal heavily with jet lag and jet lag avoidance and shortening of jet lag after somebody's changed multiple time zones. In this conversation, we've been talking about more mood and seasonal affective disorder, things like that. Why is it that the product marketing seemed to emphasize so heavily the anti-jet lag effects?
We are projecting light into these photosensitive areas of the brain, and these photosensitive areas are activated by human charger. And what the activation means is we get these chemical compounds which are natural, like serotonin, dopamine, and noradrenaline, which are released. And that is kind of leading to increased energy levels, improving mood, mental alertness, and then reducing the effects of jet lag. And maybe the reason why there's so much of the jet lag is that we did a really nice trial in jet lag some time ago, 1.5, 2 years ago. And we were so proud about it, so we wanted to put it in front. And going that jet lag route, it was pretty much like based on our own and our colleagues and our friends' initial observations that when they were using this bright light therapy device while they were on a business travel or traveling for leisure, they didn't have the jet lag. So we did a study about it and the results turned out well. So we are able to alleviate the jet lag symptoms.
Do you have additional design iterations in the works for the human charger, or is this a case where the technology is already doing what you intended and maybe meeting what you expect the science tells us is probably possible?
What we are right now working on is what we call human charger version 3, which would be like any headset that you plug into your smartphone. So we could have this podcast via that headset, so it has the voice integrated into it and then connected to the app that would kind of automatically dosage the needed amount of light for you. The problem with our modern lifestyles is basically that we are too much inside, we are too little in sunlight, we are too little outside, and this type of a normal office lighting, it's not really on the necessary intensity level that you should have. I mean, if you have the whole of your day in an office, you are not really getting the light that brain thinks that you should be getting, and that's a big problem. So what we did with Human Charger, we had a test protocol where we kind of changed and altered this waveform or this spectral curve in a way that what is the right combination of the blue, red, and green light areas. And then we have an idea that what type of spectral curves are stimulating which parts of the brain. And that's what I think that if we would think about like 20 years ahead when everybody is using this as a natural therapy for even like into the more severe cases, getting more energy, which the current product is for, the spectral curve might be totally different. This is so under-researched area. There are even light therapy device researchers who are well-established, acknowledged, and I really like a good researchers who have been doing first a trial, for example, post-traumatic stress disorder, which is cured with bright light very effectively. They do one trial with the one set of bright light lamps, and I mean the traditional light boxes. They get tremendously good results. Then they do the next study and they get a bright light manufacturer to sponsor it. They get different results. And then they're wondering that how this could be that I wasn't able to show the effect in this bigger study. And it is because they have changed the light spectral curve. And we actually realized this almost by accident in one of our earlier trials where we didn't realize still yet that, hey, it really plays a difference. The brain is really sensitive to different spectral curves. And we just kind of bought and kind of constructed devices based on this like luxes that you define, the standard measure how you define the kind of a light power. And we didn't realize, then we kind of got that, hey, why did this group of patients, individuals react to this really well? And they didn't react, although they are very similar patient groups. And then when we took the devices, we realized that when we measured the similar like a lux-defined LED lights, they were totally different spectral curves. And then we figured out that this spectral curve, and that started the huge matrix of different indications and different spectral curves and that type of testing.
The way that we're doing it in our studies is all through the eyes, but it's not through the visual system, though. What's fascinating about this whole process is that there are different kinds of receptors in the eyes. Some of them are involved in vision, and other ones we actually didn't know about until about 10 years ago that they even existed. And these are basically called intrinsic photoreceptive retinal ganglion cells. And these are a type of cell that there's quite a few of them in the retina of the eye, but only some of them seem to be responsive to specific wavelengths of light, especially blue. So it's the blue wavelengths that stimulate these melanopsin-sensitive cells, and they don't project back to your occipital cortex the way that the rest of the visual system does. We all went through school. We all heard about the rods and the cones. That's how we see things. You know, the rods and cones take that information and send it back to your occipital cortex, and then the occipital cortex turns it into a visual image. But about 10 years ago or so, it was discovered that there's this third type of receptor, and these receptors project to other parts of the brain. And one of the prominent projections is to the suprachiasmatic nucleus of your hypothalamus, which is basically the regulator of your sleep-wake cycle. And so that led to the whole idea that maybe that's what this system is for, is to help entrain and regulate that circadian sleep-wake pattern. And when people are exposed to light, and most of our research is focused on blue, we see that it stimulates these areas, it affects the suprachiasmatic nucleus, and then affects how the pineal gland basically produces melatonin. There could be other effects of light as well. There's some evidence out there I haven't done research on, but other investigators have looked at red light, and there's some evidence that red light may actually have some alerting effects in certain ways. We've been also looking at the non-melatonin aspects of light recently, and my postdoc just recently published a paper looking at the effects of blue wavelength light on direct activation of the brain without going through the melatonin system in a sense. What we did in that situation was we brought people into a room and we had them sit in a darkened room, and they just got shined either an amber-colored light or a blue-colored light for half an hour right onto their visual system. Then we took them out of the room immediately and put them into a brain imaging scanner and did fMRI scans while they engaged in some different kinds of working memory tasks.
Yeah.
And what we found was that just being exposed to that blue light half an hour ago was enough to still enhance brain activation within an area of the brain called the dorsolateral and the ventrolateral prefrontal cortex. So these are areas that are involved in working memory and behavioral control. They turned on more when people engaged in this task if they had the blue light than if they had the amber-colored light.
Did you have a control group that got no special light whatsoever?
We didn't use any with no light. We just gave them the amber as a control because we assumed that wasn't going to have anything. And in fact, it didn't. So just sitting there with this amber color didn't seem to do anything to the brain. By the time they got in the scanner, it had no effect. And then we correlated that with how much activation activation was in the brain with their performance. And we found that the greater activation in the blue group in those prefrontal areas was actually associated with better performance. So they were able to solve the working memory problems faster and more accurately in combination. They did more correct items per minute if they had received the blue light than if they had not. Some of our other sleep deprivation work focused in on risk-taking behavior. We looked at moral judgment during sleep deprivation.
The moral judgment, I think, would be really interesting, and maybe breaking apart what moral judgment is versus just judgment in general, because those can be fuzzy terms.
Well, we basically utilized somebody else's task. So there's a person out at Harvard University. His last name is Green, and basically he had done a number of studies using this particular moral judgment task. What they did basically is they showed people 3 different kinds of dilemmas. Non-moral dilemmas, trying to decide if you should take bus or the train to get to work. And so you just have to make a decision. There's no moral component to it, no emotional component to it. It's just a cold cognitive decision. Or you have a moral kind of dilemma. And the classic one that I always tell people about is what most people have heard of in their philosophy class.
Okay, so this is that moment that I warned you about where for about the next minute, he's going to be explaining the trolley problem. If you're already familiar with the trolley problem, you can probably hit fast forward on your podcast app for a moment. And if you're not, it is super interesting and worth learning what it is, so just hang in here.
The idea is that you got this trolley speeding down the tracks and it's going to run 5 people over and it's going to kill those 5 people. Thankfully, you're just standing by the side of the railroad track next to a lever. And you can pull that lever and it's going to cause the trolley to shift onto a second set of tracks where there's only one person standing. If you do nothing, the trolley is going to kill the 5 people. If you pull the lever, it will veer it off and it will save the 5 people, but it will kill the 1. So the question is, what's the appropriate response in that situation? What should you do morally to do the right thing? Most people would say that they would pull the lever. It would kill 1 person, but it would save 5, so morally that would be the right decision. This particular task that Green had come up with also had a third condition, which he called a moral personal dilemma. Basically, it's an up-close and personal situation that's much more emotionally engaging. And what's happening now is you got the trolley coming down the tracks again, heading right toward those same 5 people, but this time you're standing on a bridge overlooking the tracks. And right in front of you is this very large person. And if you walk up behind that person and you push them off the bridge, they'll fall to their death, landing on the tracks, stopping the trolley, and saving the 5 people. So the question is, would you push the person to their death in order to save the 5 people? And most people, when you ask them that one, they'll say, no way. I wouldn't do that. I wouldn't go that far. I wouldn't push a person to their death. And so the philosophical question is always, why do they say yes in one situation but no in the other when it's the same outcome? What it comes down to is the emotional intensity of the situation. The bridge situation, you have to put your hands on another person. That's a very emotionally engaging situation, whereas the other one, you're very distant. It's like dropping a bomb on a foreign country from 30,000 feet is very different than blowing a bomb up where you can see it happen to the people. So we used all 3 of these dilemmas and gave them to a sample of military personnel when they were well rested. And then we tried it again after they'd been awake for 2 full days without any sleep at all. Basically, what we found was that sleep deprivation really had no effect on the non-moral dilemmas, that you could decide whether to take a bus or a train to get to work just as quickly when you were sleep deprived as you did when you were well rested. So sleep deprivation doesn't necessarily impair all decision making. But when we look at the moral impersonal dilemma, the pulling the lever, what we also found was people were just as good at doing that when they were sleep deprived as they were when they're well rested. So it didn't affect that either. What it did affect was the moral personal dilemmas, the emotionally charged dilemmas. In that case, people took several seconds longer to make the decision in those situations, and they were much more likely to push the person off the bridge when they were sleep deprived than when they were well rested.
Interesting. And I guess we kind of view that as making the tough decision, the hard call.
The Sophie's Choice. We actually had some things that actually were those Sophie's Choice kind of things where there were terrible decisions that people had to make. Do you smother the baby to save the village from the marauders that are coming over the hill? Horrible decisions that people would have to make. And many of them actually got very irritated taking these tests because they didn't like even having to answer those kind of questions. But we felt that it was so important that we really look at the effects of this because it could have implications for police officers, for military personnel. When you're sleep deprived, there's a chance you may make a different decision than you would have had you been well rested.
It's interesting, though. I mean, that almost makes you wonder if there might be like a hidden therapy in there for people that feel like they're typically too wimpy in their choices and that they wish they could be the tough as nails person. Is sleep deprivation a way of sort of nudging yourself towards that more tough as nails mentality?
It could be, because what we're finding also that same study was also related to emotional intelligence too. So again, it taps into the emotional side of things. We found that the people with high levels of emotional intelligence, above average levels, they didn't change in their emotional choices. They were the moral choices. They made the same decision either way. But it's those who had lower levels of emotional intelligence, they actually changed their positions more in those situations. It's like they couldn't really rely on their emotional intelligence to help them through it. And so, it's a possibility. I've never tested it, but maybe it might actually give you the ability to stand up and say something when you need to. Part of it is because your prefrontal cortex just doesn't inhibit a lot of what you're going to do in those situations too.
What do you want to study next? What are some of the nagging questions for you about how we sleep and how we might sleep better?
We've got a couple of different directions we're heading right now. We've had some ongoing studies where we're using that light therapy that I was telling you about to actually try to help people recover from brain injuries. We've only just started working on it, so we haven't published our results yet. But the idea is that if you're getting better sleep, it helps you to actually clear out some of the neurotoxins in your brain. There's some evidence that it can help to enhance development of myelin again within the brain. So what we wanted to do was to help people to recover faster by using these light therapies to re-entrain the circadian rhythm, thereby improving sleep and perhaps actually bringing about some changes in brain structure and brain function. So that maybe if they've had a concussion, this might actually help them recover faster. And some of our preliminary data that we haven't published yet, but is very exciting, is starting to show that there are some brain changes, both structurally and functionally, when people go through about 6 weeks of treatment with some of these light devices. It's helping them sleep better a little bit, but in particular, it seems to be leading to some structural changes within the brain that we're hoping are being associated with actual recovery. And then we've got another study that we're hoping to get started up with in the next few months, where we're going to be looking at basically stress responses when people are sleep deprived. And we're going to be putting them through an overall stressful situation as they stay up throughout the night. And we're trying to see if we can use personality traits to predict who's going to be able to function better under stress in those kinds of situations. A lot of these are because we know these are problems that our service members are dealing with when they're coming back from their deployments. And our overarching goal in all the work that I do is really to try to take care of our service members who've experienced either exposure to trauma or a concussion of some sort. And so PTSD is a big issue that we're trying to work on right now. And one model of PTSD, not the only model, but one, is the idea that some of what has happened may be related to a conditioned fear response. I wouldn't say it's necessarily the best model, but it's one that we're working on. And so what we're looking at is, can we use a fear conditioning experiment and then essentially extinguish the fear response that a person has, follow it up with the light therapy and get them to sleep better. And our hope is that sleep is actually going to help consolidate and generalize that fear extinction memory more effectively for those people with post-traumatic stress. So we're testing that theory out now to see how well it plays out.
Smart Drug Smarts.
So thank you so very much to both Dr. Scott Killgore and Timo Ahopelto for taking the time for those interviews. Lots of different topics covered. Some really interesting and far-flung ideas in there. The apparent ability of introverts to deal with lack of sleep better. Sunlight being able to actually make it through the human skull and get to the brain where it has an effect. Also, I felt like we should have put on alarm sirens at that one warning moment to draw a double underline on the fact that when you get sleep deprived, the amount of sleepiness that you actually feel maxes out pretty quickly, although your performance degradation can continue. So you're getting mentally foggier even though you don't necessarily feel that you're getting mentally foggier. So the human charger is made by a company named Valky, and the fine folks at Valky were nice enough to give us a couple of human charger units, never before used, never seen the inside of a human ear, for us to give out to the Smart Drug Smarts listening audience. This could be you. And what I'm going to do, since it's just the start of the month of April, and you guys know how fond I am of iTunes reviews, is we're going to wait until the end of this month, look at all the 5-star reviews that have come in over the course of the month, and then randomly select 2 of them, and those 2 will be sent a human charger unit. Valky ships internationally, so it doesn't really matter where in the world you are, but you will need to send me an email with a screen capture of your review and say, hey, this was me, and also send me your name and physical mailing address. My email is jesse@smartdrugsmarts.com, and I'll put all the details about this up at smartdrugsmarts.com/175. That'll be the post associated with this episode. And we'll also put in a little how-to. If you've never left an iTunes review before, you're not exactly sure how that works. It's really simple. You could figure it out, but just in case, we'll have a little tutorial up on the website also. If you really want a human charger and you cannot wait for the end of the month, or you're not feeling feeling particularly lucky or you just hate leaving reviews, we'll also have a link over to humancharger.com that includes an affiliate tracking code so they'll know that you came from us, and we'll also get you a bit of a discount off the normal Human Charger retail price. So all this and more at smartdrugsmarts.com/175.
Smart Drug Smarts, ruthless listener retention gimmick.
So I feel like there's a parallel to be made between World War I and the drug war, both of which either went on or have gone on for a long, long time and have kind of calcified into long-term trench warfare with neither side gaining or losing much ground, or if it is, it's quickly recaptured, reconquered by the other side, and things stay essentially as they are despite leading to misery and frustration, but not much progress. Of course, we know how World War I ended. We don't yet know how the drug war is going to end, but in any war, one of the obligations of the participants is not just to put people in uniform and give them weapons, but also to convince the populace that this is a fight worth having, a fight worth expending lives and resources on, and so having the necessary propaganda on hand to kind of rally up your own people. In the case of the drug war, we've had these catchy slogans like "just say no," and both the implicit and often explicit ideas that recreational drug use predictably has bad outcomes, often very bad outcomes. Sometimes these assertions are backed by evidence, sometimes they're not, but the propaganda very frequently doesn't make that distinction. And sometimes the science can really slap the propaganda in the face. There was a research study that showed a relationship between a high childhood IQ and a wide range of illegal drug use among adolescents. A positive correlation, not what Nancy Reagan would have you believe. Of course, this was a correlative study, not a study looking at causality. A study published in 2011 looking at the 1970 British cohort study. This was over 16,000 babies born in April of 1970. They found there as well that high IQ scores at 10 years of age were linked to marijuana use at 16 years of age, as well as marijuana, cocaine, amphetamine, and ecstasy use at 30 years of age. And just recently, another longitudinal study looking at 6,000 British youths who were studied and followed up with a total of 7 times over 7 years, starting at age 11, found that academically successful children were more likely to drink alcohol regularly and to smoke marijuana versus their less academically successful peers. These kids were actually twice as likely to drink alcohol regularly. This was defined by the study as being drunk more than once weekly or 52 times per year. And while the high-performing kids were less likely to smoke cigarettes, they were more likely to use marijuana. And contrary to the idea that this was just youthful experimentation, the likelihood that their use would continue persistently over this 7 years of repeated testing was double that of their less academic peers. Again, just a correlative study, so nothing here is indicative that alcohol or drug use is causing these kids to perform well academically, but it might be that these intelligent and well-educated kids are more likely to recognize that the risks associated with consuming drugs are often widely overstated, especially in sort of ham-handed propaganda. Since there's every reason to believe that exposing young brains to drug use, especially things like being drunk 52 times per year, is probably not going to be optimal for the brain, all the more reason why keeping the propaganda backed by hard science and good evidence and not overstating claims is probably going to be a lot more effective than cartoon scare tactics for actually getting kids to make the right choices.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, so you heard it. That is all for episode number 175. Thank you for hanging around until the end of this one. Bit of a longer episode this time, but hopefully well worth it. If you want to see the links to anything that we talked about here, you can find all of those up at smartdrugsmarts.com/175. And hidden in that URL is the secret decoder ring to find any of our episodes. They should all be up with the episode number, basically getting you straight there. Last week, for example, was episode number 174, when I spoke with Dr. Adam Galinsky about creativity and its relationship to sarcasm. And next week, back by popular demand, we're going to be having another psychedelics episode, talking with one of the primary researchers in the field, a major mover and shaker for the last several decades, and certainly one of the main people that can take credit for the current psychedelic research renaissance going on, Dr. David Nichols. That'll be coming at you next Friday, so mark your calendar, be looking forward to that. But in the meantime, have a great week, get entered in that giveaway contest I talked about earlier, and above all, stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.